In order to test our hypothesis, we'll perform a comparative analysis of the gene sequence of the toll-like receptor and nod-like receptor of the tumor-infiltrating dendritic cells with and compare it with the DCs in the lymph nodes. We will also investigate the expression levels of the toll-like and nod-like receptors in the tumor-infiltrating dendritic cell as well as the DCs from the lymph nodes with and without oral administration of Bifidobacterium longum. Finally, we'll test if mutation in NLR and TLR gene could affect the anti-tumor immune response by the Bifidobacterium. We'll perform similar in vivo experiments explained in AIM-1. At the endpoint, 3 weeks after tumor engraftment, we would excise the tumor and extract the late tumor-infiltrating dendritic cells. We would also extract the dendritic cells from the lymph nodes as a control. We would then perform sequencing of the TLR and NLR. We would also quantify the IFN gamma production. Uh, we would do this by performing a co-culture of dendritic cells with the Bifidobacterium longum and quantify the IFN gamma production. Uh, to understand the gene activation profile of toll-like receptor and nod-like receptor and their downstream proteins, we will quantify the steady-state mRNA level by RT-PCR. And to pinpoint the key player in the immune response, we will, based on the previous experiment, modulate their expression using siRNA or plasmid overexpression. Altogether, we hope to understand if there is any modification in the genetic sequence of the pattern recognition receptors in tumor-bearing mice when compared to the wild type. We will also identify if IFN gamma is a key player in the activation of anti-tumor immunity, and if this activation happens via zeta of the nod-like receptors. If we do not find any relevant modification in the pattern recognition receptor, we will look at PDL1 gene activation and expression in tumor cells. PDL1 is a ligand that has an immune inhibitory activity, and it's highly upregulated on tumor cells from a broad range of cancer types. So we would like to see if uh, the PDL1 gene activation is affected by the translocation of Bifidobacterium longum to the tumor site from the gut.